Good afternoon and welcome to the USDA Human Nutrition Research Center on Aging's Monday Seminar Series. Before introducing today's speaker, let me remind you to ask your questions for the speaker using the Q&A icon located on the webinar toolbar. Please do not use the chat box. You can add questions at any time during the presentation as they arise. There is no need to wait until the end. Each month this year, our seminar series features a speaker from the different ARS Human Nutrition Research Centers. Today's speaker is Dr. Linda Larson Pryor, who is a faculty member and director of the Budding Brains Lab at the Arkansas Children's Nutrition Center. Dr. Larson Pryor is also the associate director of the Center for Childhood Obesity Prevention and professor of neurobiology and developmental sciences, neurology, pediatrics, psychiatry, and biomedical informatics at the University of Arkansas Medical Sciences. Dr. Larson's research focuses on interactions between the brain and the environmental factors such as nutrition and sleep across the lifespan and in both health and disease. And today, Dr. Larson will discuss her research in a presentation titled The Role of Sleep and Cognitive Function Across the Lifespan. And with that, I'll turn the microphone over to Dr. Larson Pryor.
Thank you very much, Paul. Thank you for that nice introduction. Hi, everyone. It's really nice to be here and to be able to talk about one of my favorite subjects, which is sleep. So we're going to just get started by talking about what sleep is. So first, if I can make my cursor work. In there. All right. I commented that I don't like these pointers, so I guess I'm going to get rid of it because it's just being recalcitrant. So first, sleep is a fundamental rhythm of the brain. It is constrained by the circadian cycle, which is a day-night cycle of approximately 24 hours that's present in all living organisms on Earth. The central regulator of the circadian rhythm in the brain, or circadian oscillator, lies in the suprachiasmatic nucleus of the hypothalamus, which is illustrated here in that little red dot in the brain. The suprachiasmatic nucleus entrains the oscillator with external and internal signals and is responsible for a number of important functions, including the sleep-wake cycle and energy homeostasis. As I said, the nucleus, the SCN, contains an intrinsic pacemaker that has a rhythmic oscillation, but that oscillatory cycle has to be entrained, and it is entrained primarily by light, which is the primary Zeitgeber or timekeeper for the circadian cycle. It is also entrained by other peripheral signals, including nutritional status, and we're going to talk about that later. In this diagram, you're looking at the top here, you're looking at the fact that we have a sleep cycle that is constrained first by the circadian process known as process C, and second by a homeostatic process called process S, and they operate together to develop our cycle, the cycle between sleep and waking. And so you're looking here at the circadian rhythm, a clock, which is provided here by this sinusoid, versus this homeostatic rhythm, which is much more like a temperature regulator, temperature controller, in which sleep need or sleep drive increases during the day and is reduced over the course of a night's sleep. And in the lower diagram, we're looking at one in which we show how this works. We have a drive to sleep and a drive to wake. They are transmitted through the hypothalamic nuclei together with a number of brainstem nuclei. Light will predominantly entrain through the retinohypothalamic tract, and the circadian process will drive first wake, then sleep. And this homeostatic process will, over the course of wake, it will saturate and then drive sleep. In addition to the suprachiasmatic nucleus, which is the master clock for the system, Clocks are also found in the peripheral organ systems, including the alimentary tract. The timing and composition of meals has been shown to entrain these peripheral oscillators, and by virtue of the complicated interactions between them that are illustrated schematically here, also help to entrain the SCN pacemaker. Studies have found that poor sleep habits, jet lag, and shift work all dysregulate the circadian rhythms and therefore sleep-wake and feeding-fasting cycles as well. Sleep is a physiological systemic state. It's characterized by changes in neural activity, which is what we're going to focus on in just a minute, changes in the gut microbiota composition, metabolism, and of course, in system homeostasis. The gold standard for studying sleep in a laboratory is polysomnography. So this is non-invasive, scalp-recorded brain activity over the course of a night. And what you're looking at in the diagram is on the top, the characteristic electrophysiologic patterns recorded in each of the main stages of sleep. Stage 1, which is drowsiness. We all know this one. This is the one where we're all kind of falling asleep in a lecture hall. Stage 2, which is our first true sleep stage. And I hate this program. And stage 3, which is slow wave or deep sleep. And finally, stage REM, REM stands for rapid eye movement sleep. And what all I want you to do is to pay attention to the fact that the signals at the top, the waveforms that you're looking at, change in each of these stages of sleep. So they are staged by reading these EEG. And then we can come down, and because trying to read 8 hours or so of this would drive you absolutely crazy, we're going to create what's known as a sleep hypnogram, which is what you see on the bottom of this slide. And the sleep hypnogram consolidates all this information and gives you a visual representation of what's happening over the course of a night of sleep in any individual that you record. And we are looking at sleep in a healthy, normal adult. So what I want you to see over here is we start in wake. This is stage wake right here, and drop down through stage 1, which is drowsy, stage 2, our first stage of true sleep, and then into slow wave sleep, stage 3. Our longest bout of slow wave sleep occurs earliest in the evening, as we dissipate our sleep drive. After one bout, we swim back through stage 2 and enter stage REM. So this is your dreaming sleep, and you see that the first bout of REM is short. Drop back through stage 2 to 3, back up to REM, and so on over the course of the night. Notice that as sleep need dissipates, slow wave sleep dissipates as well. So slow wave sleep starts out to take up most of its time early in a sleep bout, and by late in a sleep bout, we're only in stage 2 or dreaming sleep. Whereas REM does exactly the opposite. We have very little REM sleep early on, and we have a lot more REM at the end. And so this kind of hypnogram allows you to look at that kind of patterning. This is the characteristic night of sleep in a healthy young adult. And most adults will cycle through the night several times, somewhere between 2 and 7 cycles per night. And what defines a cycle? One shift from stage 2 to either stage 3 or staying in stage 2 and back to REM. So you can kind of say each of these REM cycles is going to define a cycle of sleep. This is different in infants. So we're looking here at polysomnography in an infant around 2 months of age. And you can just look at this and say, oh, those waveforms are different. In infancy, the 4 sleep stages that you saw in adults don't exist. We have a wake state, which they don't spend much time in, a state known as active sleep. Actually, in infancy was the first place that REMs were ever discovered. So this is a period of rapid eye movements. There is reduced electromyography in the chin, and respiration becomes somewhat irregular. Then we have a transitional period, and finally we have what's known as quiet sleep, which is akin to the non-REM sleep in an adult subject. We're going to revisit the difference between sleep patterning in infancy and early childhood relative to adulthood and old age as we go through this talk. But let me just begin by saying, you can look at the hypnogram. They do cycle, but they spend a lot of their time in this REM or active sleep period. They also spend a great deal more time asleep than an adult does. So much less time in wake, much more time in sleep. And that time spent in REM reduces over the course of the lifespan. And you notice that non-REM sleep, and this includes both stage 2 and stage 3, non-REM sleep also increases, is high in early childhood and reduces somewhat over the lifespan. Of course, sleep is also a behavioral state. Normally, sleep alternates with wake, but it is very strongly influenced by modern lifestyle. So here's the things that we can change behaviorally. How long do you sleep? Do you wake up to an alarm, or do you sleep as long as you want? Do you get 5 hours of sleep because you stayed up and partied all night and then had to go to work? Or do you go to bed at 9 o'clock and get up and go to work? You get to decide that. That's a behavioral decision. What about when you sleep? Do you sleep during the day, or do you sleep at night? If you're a shift worker, you may end up sleeping during the day. But we've just said that the circadian cycle is off if you do that. But it's a behavioral choice. And of course, where you sleep is also your choice. You can see that one might be a little bit more comfortable and more conducive to a good night's rest with all of those 7 cycles in there than the other, but this is a behavioral choice. Well, okay, now we've talked about what sleep is. What about why we sleep? What do we know about the reasons that we sleep? Well, right at the moment, and actually for some time now, since 2003, the most influential hypothesis of sleep is that first proposed by Giulio Tononi and Chiara Chieli, called the synaptic homeostasis theory. This proposes that the role of sleep is actually in neural, cellular, and synaptic function. And it says that what happens during waking period is you're learning things, you're thinking, you're taking in information from the environment, and the brain is taking all of this in, and it's plastically remodeling to take advantage of what you've learned that's new, what you've gained in terms of information, good or bad, from the environment. It's putting all of this together, and it's doing so. It instantiates all of that in the synaptic signaling and hormonal signaling that make up its communication system. And it gets overloaded. So when do you sleep? When the brain has to reset. So you go to sleep, and those synapses have a chance to clean themselves up, put everything back in order, and then you get to start over again, nice with a clean bill, a clean sheet for the next day. Okay, so that's one idea, and it's perhaps the most pervasive right at the moment, and it has been tested a number of times. Giulio was in my lab one day, and he said, "Please prove me wrong." And so far, nobody has managed to do that. We're looking at the cellular level. Well, what about at the behavioral level? Well, we know that when we look at kids, as I said, they don't consolidate our sleep. They don't sleep like an adult. And they need a lot of sleep. So in young children, most of them take naps. Why do they take naps? We said sleep need goes up and we go to sleep, and then we wake up after everything's been dealt with. But little kids don't do that. They nap. Well, why do they do that? Well, sleep pressure probably does play a major role, but one theory is that a major function of these maps, in fact, a major function of sleep per se, is in the maturation of memory networks. So that little kids, all of the networks that are considered sort of primary networks in the brain to do the things we all have to do, learn to talk, learn to walk, eat, drink, all of those things, those networks are in place, but over the course of development, they are very, very much refined and they grow. And one of the ones that's going to grow and become refined is the memory system. So the suggestion is that napping in early childhood results in a more efficient memory system, and particularly in storage. So one idea is that, and this is true in adults as well, it's been shown that a power nap in the afternoon can improve learning and memory later in the day in adults. In fact, in adults, research has indicated that sleep selectively benefits memories for emotionally salient information. In this PNAS study, a very large population was used to show that this is true. So we're looking here at the more salient stimuli showing the greater benefit from sleep relative to things that were less salient. We also have a model that looks at neurovisceral integration. This proposes that changes in autonomic activity, and in particular using heart rate variability as an index, are related to prefrontal control over regions that support cognitive function, such and include emotional regulation, twice even. So when parasympathetic tone at rest is high, it's related to working memory performance and improved working memory performance and inhibitory control. And of course, we get an increase as well in non-REM sleep. In another study, an interesting model was proposed by Chen et al., who suggested that this slow oscillation switch model, in which long-term memory and working memory or short-term memory are supported separately during slow wave sleep, so that long-term memory is dependent more on hippocampal ripples, which are known to occur during REM sleep and to help consolidate memory. That long-term memory is more dependent on that kind of activity, but short-term memory or working memory is supported by the slow wave functions alone. So one reason that is popularly, I'm not going to say popularly, one reason for sleep that is pretty much stood pretty much stood up to all the tests that have been made and has been shown repeatedly by a number of different laboratories, is that when we sleep, we learn better, we think better, and we're more alert during the day, more available to make good decisions based on the data that we're taking in during the day. All right, so sleep is good for us. How much do we need? Everybody's a little bit different. So human adults sleep between 4 and 10 hours a night on average. There really is no number that I could say to you, oh, this will be perfect for you. This is exactly where you need to be. But if you look here at our population averages, 4 and 10 to 12 are on the ragged edges. The mean is around 7 to 8 hours. Lots of studies have shown that short sleep, which is less than 6 hours of sleep on a regular basis, is very bad for you. Interestingly, some studies have shown that long sleep, which is defined as over 9 hours, is also not so good for you. The data there is a little less clear. It's quite clear for chronic sleep restriction. And in a minute, we'll talk about that. And the lower diagram here simply reiterates what we already learned, which is sleep, the number of hours needed for sleep on average at an age differs over the lifespan. Much higher in infants and young children than in adults. So that's how much we need, but what do we normally get? More than a third of US adults simply don't get enough sleep. So 23% of US adults reported getting less than 6 hours of sleep a night. And that's on average. Many people get 6 hours on average and 4 or 5 a few times a week. 7 to 8 hours, still the average, but a lot of people simply don't get enough sleep. And what does that mean if you don't get enough sleep? Well, first of all, if you don't get enough sleep, you tend to be sleepy during the day. And if you're sleepy during the day, you're not alert or paying attention. So you're going to make more mistakes. In fact, sleepiness during the day has been linked to major league disasters, such as the Exxon Valdez crash, Chernobyl. All of them are believed to have been due to a sleepy technician on duty. So if you don't sleep well, or you don't get enough sleep, you have poor cognitive function, you have the probability of taking in more food. We eat when we're hungry, when we're tired, and an increased risk of disease. In fact, a lot of studies say that chronic sleep restriction has increases morbidity and mortality across the lifespan. So we're looking here from the CDC's report, looking at difficulties in concentration, remembering, driving, which is not a good thing to be sleepy during, and doing your financial planning. Maybe not just yours, maybe your business's financial planning. So the consequences of chronic sleep restriction, which is not uncommon in adults and increasingly not uncommon in children, are actually dire. Okay, so what happens when we sleep to our brains? We've said that this is a process for our brains. We've suggested that there's the homeostatic need for sleep that may have something to do with the brain's communication and operation. So we know that during about a night's bout of sleep, the brain actually reduces the energy it uses. And so you see here in a study done in Brown in 97 and replicated by Maquette and his colleagues in 2012, during wake, energy usage is very high. It's reduced in stage 2 sleep, significantly reduced in slow wave sleep, and then brought back to about the level of stage 2 during REM sleep. Not as high as wake for the most part. So we know that the reductions in energy consumption are greatest in those regions that are involved in attention, cognitive function, and cognitive integration. One of the findings, one of the reports from Brown et al. Was that, in fact, there was a continued reduction in energy utilization once someone woke up in the daytime, and then it increased again over the course of the day. So we attempted to replicate that study in a pretty large study looking at whole brain metabolism. So in the top right at B, we looked at glucose metabolism. In C, we looked at oxygen metabolism, and we looked at cerebral blood flow here in D. And as you can see, during the morning, relative to at night, these are all in FDG PET. During the course of the day, at the end of the day, we measured, and then again, early in the morning, being very careful not to let people walk around. So we wheeled them to the imaging center. And we could not replicate the report of reduced energy expenditure following a night of sleep. Instead, we saw no change. As soon as they woke up, they were right back to their normal waking states. However, we do know that the change over the course of a night's sleep does occur. We know that now people have become interested in this, and so we have, as probably most of you know, we have a number of very large studies that are being funded by the national funding agencies in multiple places. And these data that we're looking at right now are from the UK Biobank study of sleep, age, and executive function. So this is from the Oxford group, largely the imaging centers at Oxford. And right at the moment, at the time this paper came out, there were 479,000 individuals between the ages of 38 and 73. And they had imaging data, MRI data on about 38,000 of those people. And so what did they find? They found that the median daily average for sleep was about 7 hours. And they found that these individuals showed, here's imaging, they have significant imaging data on all of these. What they discovered was that sleep duration tended to remain stable over time. And it ranged between 2 and 14 hours, about what we said, with the optimal functioning seen with sleep durations of about 7 hours per night, but 6 to 8 also provided excellent function. So that's what you're looking at here. Here's the duration. And then this is the executive function score. And you can see it's high for our range of 6 to 8, and then falls off rapidly with decreased sleep and increased sleep. So again, supporting that idea that too much and too little, both not so good. In addition, they found differences in the brain volumes between individuals who slept this optimal period of 6 to 8 hours versus any of the outside banks. And they looked specifically for what areas of the brain indicated they had greater volumetric volume in these good sleepers. And those were in frontal, hippocampal, and cerebellar volumes. And so what does that mean? Frontal cortex is your executive function cortex, your decision-making cortex, your control cortex. Hippocampus is your memory cortex. Cerebellum is everything. It's got all of those things plus a fairly strong motor control component. They found in mediation analysis that brain volumes mediated the effect of sleep duration on executive function. So you found these increases in volume, they found increases in the ability, the executive function abilities in these individuals, and mediation analysis showed that that was dependent upon increased volume in these important regions of the brain. I'm going to pound this home, can you tell? Chronic sleep restriction also impairs memory. So in addition to looking at people in the wild and asking what their propensity, their habits are, which has the problem of all self-report, we can also bring them into the laboratory and impose sleep restriction. So in this study, individuals were brought into the laboratory. They were given a 9-hour sleep opportunity on the first 2 nights. We have a known effect of sleeping in a strange bed. So you bring them in, you got to give them at least 2 nights to acclimatize to where they are. On the 3rd night, in the experimental group, there were 1, 2, 3, 4, 5 nights of restricted sleep, in which people were asked to sleep only 5 hours a night, and the control subjects continued to sleep their normal, get their normal 9-hour sleep out. At the end of that period, they did a working memory task. It's called an NBAC working memory task, and it basically says, I'm going to ask you to look at a series of images, or, well, usually images, and you're going to tell me whether the image you see is the same as the image you saw 1 image back, or 2 images back, and that's the N, 1 back or 2 back. We know that the more backs you go, the harder the task is. So the normal is to do a 0 back for a control and then a 2 back to actually test for working memory performance. And what did you find? You found that those individuals who were sleep restricted simply did not do as well. Their accuracy in remembering, just bringing in the information was lower. Their ability to recall the information was impaired. It took a lot longer. And their accuracy in retrieving information from memory was also impaired. So we already talked about the fact that it looks like sleep restriction will impair memory. This was only 5 days. What if you did this for 6 years? We also know that sleep restriction, sleep loss is associated with neuropsychiatric diseases. So sleep disturbances are core symptoms of a number of psychiatric disorders, including major depression, bipolar disorder, PTSD, and generalized anxiety disorders. Psychiatric symptoms, in turn, are associated with several primary disorders of sleep, and that includes sleep apnea, narcolepsy, and insomnia. So we can see in depression, we can see either hypersomnia or insomnia. Same with bipolar, which is, and then in schizophrenia, normally insomnia, and in many cases, sleep apnea, sleep disordered breathing. In ADHD, interesting, restless leg, sleep disordered breathing, and a number of parasomnias are very common. So a number of sleep disturbances related to psychiatric disorders. In some instances, particularly depression, this looks to be a bidirectional process where sleep restriction increases depressive symptoms and depressive symptoms increase the amount of sleep disturbance. So we're going to look at that. Here we're looking at clinical depression, and in this case, hyposomnolence or insomnia. At the top, we see the polysomnogram of a good sleeper, and in the bottom, we see a patient with major depressive disorder. And I'm just going to point out, here's stage wake is at the top, stage REM is in red, as you were seeing before. And stage 3 is at the very bottom. So you're seeing this is blue. Stage 2 is what's in green. So what's happening? You're almost seeing a reversal in major depression where they're awake longer. This is definitional for insomnia. They drop to stage 2, not stage 3, come back to REM and oscillate through wake, go back to stage 2. So you notice there's absolutely no stage 3 sleep in this somnogram, in this hypnogram at all. They're getting REM, but mostly they're getting wake. So significant reduction in the amount of sleep that they're getting and no slow wave sleep at all. We've talked about the fact that slow wave sleep is a major player in long-term memory consolidation. Now we're going to take this, which was from non-invasive measurements, and we're going to look down here. Here's the sleep cycle. This is essentially another hypnogram. And we're going to look at neuronal activity in the brainstem. And we're looking at 2 cell types, which we know oscillate and are affecting sleep and wake. And you see the regular clear rhythms and the rhythms between these and REM sleep bouts that just are not happening in major depressive disorder. So clear disruptions of the sleep cycle at the level not only of behavior and scalp recorded EEG, which is cortical activity, but also at the level of the brainstem neurons that are helping to control that sleep. In addition to these problems, we know that there's a linkage from the brain to the gut that's related to sleep itself. So in insomnia, for example, we see alterations in neurotransmitter release, as we just talked about, and alterations in the metabolite concentrate, the metabolite composition of the gut microbiome. In obstructive sleep apnea, where we have intermittent hypoxia together with sleep fragmentation and daytime sleepiness, we see that these changes in the gut microbiota induce gut dysbiosis and leaky gut. So we're reducing the tight junctions between the chromatin cells, and we're creating a leaky gut in OSA. So, and does this affect the central nervous system? Yes, it does. There's a bidirectional communication between gut and brain that we talked about in terms of the clock and have not yet talked about in terms of the way in which those kinds of communication actually occur. We have a very interesting sleep disorder to discuss now. This is actually a sleep parasomnia, and it's called REM sleep behavior disorder. It has emerged as a very promising prodromal marker for Parkinson's disease. And prodromal, what does that mean? We know that, like Alzheimer's disease, like many neurodegenerative disorders, the neurodegenerative process that is going to ultimately result in development of classical Parkinson's disease is going to start at least 10 and maybe 20 years before any symptoms ever occur. So REM sleep behavior disorder has been shown to occur about 10 to 11 years before a diagnosis of Parkinson's disease in a substantial number of individuals with PD. And so there are studies going on now to look at people with idiopathic RBD and to determine what percentage of individuals are going to then go on to get Parkinson's disease. The prevalence of this disorder in patients with a diagnosis is also quite high. So between 25 and 60% of Parkinson's patients exhibit RBD. About 90% of people with dementia with Lewy bodies, and about 90% of individuals with multiple systems atrophy. All three of these diseases are alpha-synucleinopathies. What is RBD? It is a REM sleep phase parasomnia in which the muscle atonia that is found in normal sleep is lost. So when you fall asleep and you have a dream, and you're going into REM sleep, your peripheral musculature is paralyzed. It's not really paralyzed. It's a paresis. It's much, much weaker. So you dream and you twitch, but that's it. In RBD, that atonia is gone. And so now when you dream, particularly if your dreams are violent or frightening, you enact them. And it is the case that in this disorder, dreams are frequently violent or unpleasant. So people are terrified or angry. And the little diagram shows you, they end up taking it out on their bed partners a lot of the time. Sometimes they simply fall out of bed. Interestingly, it's not super common to add to this sleepwalking, but it has happened. So as we just said, it's most common in the synucleinopathies. It's rarely seen in tauopathies or amyloidopathies. So it's fairly unique to a class of disorders that are related to alpha-synuclein, and that is an aspect of the disorder that has not yet been terribly well studied. It can be secondary. So secondary RBD is generally associated with some common medications, actually the SSRIs, the very common antidepressants. Or it can be idiopathic, where we have no idea what caused it. And so we said is most common in synucleinopathies, but as we said, a large number of people with idiopathic RBD will develop a synucleinopathy. So whether the synucleinopathy is a precursor or just a chance correlation, or there's some causal mechanism, is not yet known. So we did an early chart study of 115 RBD cases in our neurology department, which housed our sleep clinic at WashU. And indeed found that idiopathic RBD was more common in older men than in older women, but it was very common in younger women. And in that case, it was associated with autoimmune disorders. And ours is the only paper I know of that's actually reported that. We also then looked at the relationship of idiopathic RBD to other diseases, in particular in red here, neurodegenerative diseases, those which were diagnosed after diagnosis of RBD in fuchsia, and those which were coextensive with the neurodegenerative disease. We also looked at narcolepsy. It's not uncommon in narcolepsy, and then at other CNS diseases. And you see that in men, it follows the pattern is what you'd expect. You see with increasing age, a large number of individuals exhibiting RBD, either in conjunction with neurodegenerative diseases or having the neurodegenerative disease occur after onset, before the onset of our RBD. And narcoleptics tended to be in the younger age groups. But that pattern was not seen in women. Although there was an increase in RBD plus neurodegenerative disease as women aged, you did not see the rather substantial increase across middle to older age in women. And you did see other CNS disorders, and we said particularly autoimmune disorders, being more common in women than in men. We became interested in this, so we put in a pilot study. This was actually, at the time, my resident, and now a full professor in neurology in the sleep clinic at WashU, Dr. Yoel Zhu. This was a pilot study that she did as a fellow of the Sleep Research Society. And we didn't have a large study sample. We had 10 RBD individuals, and mostly men, and 10 controls. They were all in their 50s. This is idiopathic RBD. None of these individuals with RBD had any known neurodegenerative disorders. And you can see that their rather simple memory screen showed that they were all cognitively normal. So we recruited all of these individuals from the Alzheimer's Disease Research Center and looked for people who endorsed RBD. And then we tested for RBD on a validated questionnaire, brought them in for full polysomnography. Dr. Zhu did that. And then we did neuroimaging. So this is MR imaging looking at functional connectivity in these individuals. And what did we find? We found group differences, even in this very small population, in visual networks. And these networks showed reduced connectivity, both within the visual area itself and also between the visual system and the networks associated with attention. And you can see that illustrated down here in slice by slice across the superior to inferior cortical areas. So these are areas, by the way, that we know are particularly affected in Parkinson's disease. So this is suggestive of something about this link. And as I said, it's known to be prodromal. This was a study looking at 121 subjects with RBD confirmed by polysomnography. A clinical diagnosis of RBD has to be confirmed by polysomnography. In these individuals, there were other prodromal symptoms that were considered: constipation, anosmia, orthostatic hypertension. These are all very, very common. Daytime sleepiness, erectile dysfunction in men, depression and urinary dysfunction. Constipation, by the way, next to sleep disorders, constipation is probably the most common prodromal symptom in Parkinson's disease patients. So they looked at individuals who had prodromes, and they asked how many of these individuals converted to PD or DLB from RBD. And so what you're looking at here is that those individuals with these prodromes are illustrated here in red, and there was a much more rapid conversion from RBD to PD in that population. So RBD is a prodrome, add a second prodrome, and you're much more likely, and earlier on at least, to develop overt Parkinson's disease. You will notice that those who only had RBD also converted, but not at as rapid a rate. So we talked about, so we just said constipation is another major prodromal factor in Parkinson's disease. Well, if constipation is important, then maybe the gut's important. And in fact, increasingly, that's exactly what we see. So we know that the gut-brain axis is bidirectional communication between the central nervous system and the enteric nervous system. And this is mediated in part by the parasympathetic nerves, the vagus and pelvic nerves, and in part by humoral pathways from gut to brain and brain to gut. All right, so this is just a picture looking at that bidirectionality. In this study, this is a really complicated study looking at the composition of the gut microbiota in 441 individuals that included 108 individuals with idiopathic RBD, 127 first-degree relatives of those individuals, and 36 people with early Parkinson's disease, to ask, does the microbiota composition from RBD, in RBD, give us a clue to whether or not somebody's going to develop Parkinson's disease? So what you see is a progressive decline in butyrate-producing bacteria and an increase in pro-inflammatory bacteria that was present in RBD and strongest in early Parkinson's disease. But it showed this, we'll look at the butyrate ones, it showed this nice progression between controls, idiopathic RBD, first-degree relatives, those with RBD, and those with early Parkinson's disease. So the gut-brain axis and the linkage between gut and brain are playing a role in not just the development of PD, but the interaction between these prodromes, sleep disruptions, and prodromal problems in the gut. And we've said PD is a synucleinopathy, and the first place alpha-synuclein begins to misfold is actually in the enteric nervous system, not the brain, which has led to the suggestion that there is actually transmission of misfolded protein via the vagus nerve into the brain, but that is far from a proven. It's just a hypothesis that's currently out there. All right, so as we say, we now know that the gut-brain axis does play an important role in Parkinson's disease. Constipation, as I said, is very, very common, present in about 80% or more of Parkinson's patients, strongly associated with alpha-synuclein degenerative changes. And we said this is important, in addition to looking at these changes in bacterial function, the severity of constipation has also been linked to reductions in some parts of the microbiota. And in this particular study here, mediation analysis showed that the frequency of bowel movements mediated the effect of gut dysbiosis on the likelihood ratio of prodromal PD. In other words, the amount of the severity of your constipation will result in a gut dysbiosis, and that gut dysbiosis increases the probability of developing Parkinson's disease. Now we get to the next stage in this story. So we know that in older adults, doesn't matter, cognitively normal older adults, sleep architecture changes, and in some adults it changes drastically. So you're looking here on the left at our normal young adult pattern, and now at an older adult, and you see that sleep becomes very fragmented. Now, everybody's a little bit different. Some people still sleep like this at 90. Some people sleep like this at 50. But just on an average, older adults exhibit more fragmented sleep, less time in slow wave sleep, and a little more time in REM. So moving on from there, now we're going to look at another neurodegenerative change in aging, which is Alzheimer's disease. And here there's a clear link to sleep. There's definitely a huge change in sleep rhythmicity in Alzheimer's disease, reduced total sleep time, extreme fragmentation, excessive daytime sleepiness leading to common naps, and even insomnia. We also know that in cognitively normal adults, poorer sleep quality is significantly associated with higher tau in the CSF. In other words, a prodrome for Alzheimer's disease. And longitudinally, the greater the number of sleep disturbances at baseline, the steeper decline in CSF Aβ42, and therefore the greater the probability of developing Alzheimer's disease. And so you can see here, we see circadian dysfunction, very common in Alzheimer's disease, sleep deprivation, very common, changes in neural activity, increases in amyloid plaques, of course, memory impairment, which is characteristic, and tau pathologies. So definitely showing a relationship between sleep, memory dysfunction, and brain aging. pathological brain aging. So, to summarize, we know that sleep is an essential biophysical function, homeostatically regulated but strongly modulated by the circadian cycle. The neurobiological regulation of sleep involves systems that are intimately interconnected to those controlling autonomic function, feeding, and energetics. Current research shows that sleep disturbances adversely affect cognitive function and system-wide health across the lifespan, and dysregulated sleep is an important factor in pathological aging, neuropsychiatric disease, and disturbances in energy homeostasis. And with that, I will thank you all for your time and attention. I will really thank our organizers, Peggy and Eleanor. They were absolutely wonderful, and they kept me on track and they kept me informed. Big kudos to them and to Paul for being here and leading us all through this. And of course, to my team and my sponsors. Thank you very much.
Well, thank you very much. It was a very interesting presentation. I'll take the prerogative here just to ask the first question. I'm just curious, because you finished up there linking sleep disturbances to Alzheimer's disease, but is there any longitudinal evidence? Because it can be tricky. Can the prodromal or preclinical changes that will lead to Alzheimer's be affecting sleep, or is it really sleep disturbance itself that leads to the changes that lead to Alzheimer's?
Probably the most compelling evidence that sleep changes themselves can lead to this disorder is the new work on the glymphatic system. So one of the things that sleep is acting to do is to activate the brain's lymphatic analog, which is called the glymphatic system. And that is clearing, it's been shown to clear neurotoxins, including A beta. So if you don't clear them, then they increase and they clump, and yeah, it's been shown.
All right, thank you.
Believe it or not, people with Alzheimer's disease and their relatives are unbelievably willing to help with Herculean experiments. Those were overnight studies in which CSF was collected every hour.
Well, thank you. Okay, the questions are piling up here. So let me start. Richard Albert has two questions for you. I'll give you the first and then the second. The first is, has there been research in sleep cycle homeostasis and sleep cycle synchrony among the visually impaired?
Yes. Am I really up on that? No. but there has been, yes. There's circadian rhythm disruptions in those who are visually impaired, and particularly those who've been blind since childhood, and definitely dysrhythmia. You have to recognize that while light is our primary zeitgeber, it's not the only one. And another one that does work is temperature. Of course, we control the temperature of our houses now, so it's not as good as it might be, but temperature does play a major role. So there are other signals that can be used and potentially boosted. I'm not really up on that literature, but I know it exists.
Richard's second question actually starts off with a comment: thank you for an excellent and thought-provoking presentation. And the question is a cause and effect question. Might there be a gut microbiome therapy for treatment of cognitive disorders?
Well, people are certainly looking into that. So yes, I think that's kind of a hope that we can capitalize on this bidirectionality. And we know one thing: we can't artificially, that is, drug-induced changes in sleep are not going to solve the problem. If we had mechanisms where we could, if you will, normalize the gut microbiota, might that really prove to be beneficial? It might indeed, and a lot of people are working on it.
Okay, next question is from Ed Salzman, and he asks, given the prevalence of obesity being greater than 40%, is some degree of sleep disordered breathing or other weight-related impact on sleep, example, brain aging, becoming a new normal?
Well, I think that's a wonderful question. And It's one I would really dearly love to answer. So since we have children now as young as 5 who are obese, we have children 8 and 9 years old who are obese and hypertensive, with non-alcoholic fatty liver disease. What is the long-term effect on brain aging? I suspect it's not good at all. And then the question, is sleep disordered breathing the only sleep disturbance that you see in individuals with obesity? No, it's not. Dysregulated sleep is not uncommon. It's been reported to be very common in children. I did a small study about 60 people. I did not actually see that, but I think it's an excellent question. And is it becoming the new normal? Yes, unfortunately, in many ways it is. And we have no idea what the consequences are, but I don't think they'll be good.
Next question comes from Larissa Constantino, forgive me. Are there any opportunities to intervene and prevent Parkinson's disease?
Not that we know of at this point in time, but again, people are really looking hard at the gut microbiome because the synucleinopathy occurs there first, and because of this idea that there may be a prion-like transfer of misfolded protein into the brain. There's a lot of interest in. I would say that you will see that if you tracked PubMed, you will see that studies looking at gut dysbiosis and the gut microbiome in Parkinson's disease or prodromal to Parkinson's disease have just shot through the roof in the past 5 years. At the moment, that's where we are. There's a lot of contradictory data, perhaps not surprisingly. There's a huge amount of contradictory data. So we're not at the point to be able to say we can do therapeutics, despite the fact that some people would love to say that. But I think ultimately there's the hope that will provide some kind of therapeutic window, yes.
Our next question is from Michael Lustgarten, and he asks, when considering that Faecalibacterium is a soluble fiber degrading bacterium, would you expect a high soluble fiber diet to positively impact the gut-brain axis in RBD?
Well, that's a great question. Perhaps. I mean, like I said, I think dietary factors are ones that the neuroscience community is just barely coming to grips with. And to be fair, they're coming at it from the microbiota side, more of the genetics and proteomics side, and less the diet side. But I think that, I think we're still in that infancy stage of, we should all do probiotics. But yeah, I mean, you might expect that would help.
Okay, and
let me add, certainly Parkinson's disease patients play with this. They report things where when you go into the literature, there's a little bit of evidence that what they're telling you that works for them may indeed work well. So, I think that's something to keep in mind as well. Sorry.
And the final question, unless we get one more in the next minute or two, is from Thomas Chen. He thanks you for your studies, and he states, I'm a 60-year-old man waking up 2 or 3 times a night to pee. Any relationship to sleep disruption?
No. So that was why I made the point that when you look at sleep fragmentation, you get up a couple of times during the night, that doesn't make the grade. We're talking about people who are probably waking up 10, 20 times a night. That is to say, they barely go to sleep and they wake up again. So you don't need to worry about, I mean, indeed, you at 60 versus a 10-year-old child, well, that's not even fair, a 12-year-old child, the kid's going to sleep longer and more soundly than you are. But if you get up a couple of times a night, don't worry about it.
Okay, and let me just check see if there's any more questions. Another question just came in. So, just a thank you from Thomas Chen. All right. So with that, I think I'd like to again thank you for your really fascinating presentation. That I guess that's it.
So, okay, thank you, everybody. Have a good day.
Thank you. Bye.
Bye-bye.
Okay. well, a really wonderful presentation. It was nice, really nice and clear, and a lot of fascinating information.
Thank you.
Yes,
I love talking about sleep, can you tell?
It's important, but
One of my favorite topics. Well, thank you so much for having me, Paige and
Okay.
Eleanor. I really appreciate it.
thank you.
And as I
Happy
said, extremely
holiday.
well organized. I appreciate that.
Least we can do for all you did today. Thank you.
You bet. Take care, everybody. Bye-bye.
Bye.